When there is a heavy downpour of rain, or when we water our plants in the garden, it is the roots that absorb water for the plant. More specifically, the millions of root hairs present on the root tips absorb both water and minerals from the soil. These root hairs not only increase the surface area for absorption, but also assist in water absorption through diffusion. After water is absorbed, it moves deeper into the root layers by one of two pathways, apoplast and symplast. Apoplast is a continuous system of adjacent cell walls in a plant, interrupted only by waxy, suberized Casparian strips in the root endodermis. So apoplastic movement of water occurs through intercellular spaces and permeable cell walls. Water movement is through mass flow and is dependent on the gradient. As water evaporates in the intercellular space, it creates tension in the water flowing inside the apoplast. Mass flow is thus propelled by the adhesive and cohesive properties of water. On the other hand, the symplastic pathway is a system of interconnected protoplasms. In symplastic movement, water travels through the cell cytoplasm, while intercellular movement is through the plasmodesmata. Water movement takes place down the potential gradient. As water enters the cells through the not-so-permeable cell membrane in the symplastic pathway, water movement is slower. However, the symplastic pathway is aided by cytoplasmic streaming, that is, the movement of the cytoplasm. Unlike the rest of the root, the inner boundary of the cortex, that is, the endodermis, is impervious to water due to the presence of Casparian strips. As water molecules are unable to penetrate this layer, they move to wall regions that are not suberized like Casparian strips. Instead, they move inside the other non-suberized cells through the membrane. This water movement too is through the symplast, allowing water to cross a membrane to reach the xylem cells. Therefore, water movement through root layers is symplastic in the endodermis, and it is the only pathway that allows water and solutes to reach the vascular cylinder. Water moves freely again once it is inside the xylem and enters the xylem vessels and tracheids. These xylem elements are non-living and therefore become a part of the apoplast movement, which occurs in the non-living parts of the root. Some plants have additional structures associated with their roots to help in absorption. Mycorrhiza, a symbiotic association between a fungus and a root system, is one such example. Fungal hyphae absorb larger volumes of minerals and water from the soil because they have a large surface area. In turn, roots provide sugar and nitrogen-containing compounds to mycorrhizae. Some plants like pinus seeds cannot even germinate without mycorrhizae. Hence, they are said to be in obligate association with the mycorrhizae. However, irrespective of how water is absorbed, water moves deeper into the root layers through two pathways, that is, apoplast and symplast. When plant roots absorb water and minerals from the soil, they move it in an upward direction to reach different parts of the plant. Have you ever wondered how this upward movement of water and minerals takes place? This is made possible by a combination of root pressure and transpirational pull. Along with the water, various mineral ions from the soil are also pushed into the root vascular tissues by diffusion. As a result, the pressure inside the xylem increases. This positive pressure is known as root pressure. Root pressure can be observed in a plant by a cut stem experiment. In the early morning hours of a humid day, if a soft stem is cut horizontally near its base with a blade, drops of solution ooze from the cut stem. This oozing is caused by positive root pressure. Now try fixing a rubber tube to this cut stem. You will be able to collect and measure the rate of exudation and also determine the composition of exudates. The impact of root pressure is best witnessed during the night and early in the morning when evaporation is slow. At this time, the excess water collects as droplets around the special openings of veins called hydothodes near the tip of grass blades. This water loss in its liquid form is called guttation. However, root pressure itself does not account for the majority of water transport because it can provide only a modest push. Most water movement in tall trees takes place due to transpirational pull. The driving force behind this transpirational pull is transpiration from the leaves. Transpiration is the process of water loss through stomata in the leaves. Unlike guttation, where water is lost in liquid form, water is lost as vapor during transpiration. Transpiration can be witnessed by closing a healthy plant inside a polythene bag. You will notice that droplets of water have formed inside the bag. These water droplets are caused by the loss of water from leaf stomata by transpiration. The water loss due to transpiration pulls the water upwards in a plant stem. Did you know that the transpirational pull generated by this process of transpiration is strong enough to cause water to move upwards even in tall trees by as much as 15 meters per hour? Water transport is also aided by cohesion and surface tension. Cohesion is the mutual attraction between water molecules, and surface tension is the force acting on a water molecule traveling upwards in the xylem. Since cohesion tension helps the transpirational pull, it is also known as the cohesion tension transpirational pull model of water transport. Therefore, we can conclude that together, transpirational pull and root pressure are responsible for the upward movement of water in plants. Did you know that a fully grown tree may lose thousands of liters of water through its leaves on a hot, dry day through transpiration? Transpiration is the loss of water due to evaporation that occurs through pores in leaves called stomata. Stomata are not only the site of water loss for transpiration, they also help in the exchange of oxygen and carbon dioxide in the leaf. They remain open during the day when transpiration takes place and close at night. Their opening and closing is directly impacted by the turgidity of guard cells present near the stomatal aperture. The inner wall of each guard cell is thick and elastic. When turgidity inside two guard cells increases, the outer walls bulge and force the inner walls to take the shape of a crescent. The radial orientation of the cellulose microfibrils in the cell wall of the guard cell helps open the stomata. When guard cells lose turgor, they become flaccid, while the elastic inner cell walls regain their original shape, leading to the closure of the stomata. Transpiration is affected by both external factors like temperature, light, humidity, etc., and plant factors like the number and distribution of stomata. Typically, the lower surface of a dorsiventral dicot leaf has a greater number of stomata. On the other hand,